Hello, I'm Hannah Donner with the Collaborative on Health and the Environment. She enjoys bringing you the latest environmental health science through our partnerships, calls, webinars, science serves, publications, and social media. I would like to welcome everyone to today's webinar titled Developmental Endocrine Disruption and Neurotoxicity. This webinar is sponsored by the HEEDS Young EDC Scientist Showcase and the CHE EDC Strategies Partnership is one of our other partners. Our moderator today is Sarah Howard, founder and manager of diabetesandenvironment.org. We will leave time following the presentation for a brief Q&A session. You may type in questions to the Q&A feature available on the menu bar at the top of your window at any point during this presentation. After our presentation, our moderators will read out questions for our speakers to respond to. We will get to as many comments and questions as we can during the Q&A period. For those of you who have called in on the phone, we have posted slides to accompany today's webinar on our website. You can download these by going to healthandenvironment.org. Please scroll to the bottom of the page and select today's webinar. On the webinar page is a link to the slide. Everyone on the webinar right now is muted with the exception of our moderator and our speakers. This webinar is scheduled to last for 60 minutes and is being recorded for our call and webinar archive. With that, I'll turn things over to you, Sarah.
Thank you, Hannah, and hello, everyone, and welcome. Like Hannah said, this webinar is part of a series. This is our 5th in the series, sponsored by HEEDS, which stands for Healthy Environment and Endocrine Disruptor Strategies. And so we're featuring early career scientists in this series. So if you want to sign up for announcements of future webinars, you can go to heeds.org and sign up for the HEEDS newsletter, and we announce them there. Our next one is actually going to be December 1st, and so keep your eye out. So today, our topic is developmental endocrine disruption and neurotoxicity. And we'll have first 2 speakers and then a question and answer period at the very end. So our first speaker is Dr. Marissa Sobolewski, who is an assistant professor in the Department of Environmental Medicine and the Environmental Health Science Center at the University of Rochester Medical Center. She received her PhD from the University of Michigan and trained at the Smithsonian Institution. She holds a variety of positions, from the editorial board of the journal Neurotoxicology to vice president-elect for the neurotoxicology specialty section in the Society of Toxicology, as well as a number of leadership positions in the neuroscience and toxicology programs at the University of Rochester, where my son happens to go. She's going to discuss her research on how exposure to a mixture of endocrine disrupting chemicals during development affects neurological and brain development in mice. And then our second speaker is Dr. Katie O'Shaughnessy, who's a new investigator in the Center of Public Health and Environmental Assessment at the US EPA in North Carolina. She received her PhD from the University of Florida, and I don't have a hat. She held a postdoc also at the EPA. She's won numerous awards, including a new investigator award from the Developmental Neurotoxicology Society and the Innovator Award from the Society for Birth Defects Research and Prevention. She's working to build a diverse lab researching women's and children's health. She's going to discuss her research on how exposure to low thyroid levels during development affect the brain and potential biomarkers that could be used to screen for this developmental neurotoxicity. All right, so I think, Marissa, we are ready for you to begin.
Wonderful. Thank you so very much, Sarah, and thank you, Hannah, for that wonderful introduction and for the invitation. And thank you, Katie, for joining me and presenting your work with me today. Endocrine disruption and neurotoxicity are 2 of my favorite things, so I'm really looking forward to the question and answer section at the end also. As mentioned, my name is Marissa Sobolewski, and I'm an assistant professor at the University of Rochester. And I'm going to tell you a little bit today quickly about our work on some sex-dependent neurotoxicity that we found after developmental exposure to a mixture of low-dose endocrine disrupting chemicals. And I'm going to start by telling y'all something that most of you know, and that is that the endocrine system is the body's chemical communication system. Essentially, the endocrine system helps the body maintain stasis through change, either coordinating developmental change or responding to environmental stimuli that threaten homeostasis, to body temperature, water and ion balance, cardiovascular function, metabolism, or changes in the needs for metabolic supportive behavior. Our concern here today is that xenobiotics can target the endocrine system and disrupt this function. In its simplest definition, an endocrine disrupting chemical is any chemical that interferes with hormone activity. But given the breadth of this definition, and given the fact that the endocrine system is the body's chemical communication system that targets nearly every organ system, in fact, it's actually not surprising to me that over 1,000 xenobiotics have been recognized to have some endocrine active properties. And these compounds come from broad chemical classes, including herbicides, surfactants, plasticizers, industrial emissions, and many, many more. One of the most vulnerable windows of exposure along the human life course is pregnancy and fetal development. This is a critical stage when hormones shift from maintaining homeostasis to organizing homeostatic set points, development, and growth. And it's a critical window for both mother and child. And when experts measure our exposure to EDCs in pregnant women, exposures to EDC mixtures from broad chemical classes are the norm, not the exception. In fact, focusing on children right here in New York, as newborns, there is near ubiquitous exposure to bisphenol A and perfluorinated compounds PFOA and PFOS in newborn blood spots, albeit and importantly, at extremely low exposure concentrations. But these realities beg the question, are exposures to low-dose EDCs safe? And are they safe in mixture? Elegant research from a group of scientists focused on understanding EDC mixtures and the male reproductive tract indicate that in fact, mixtures of EDCs, even if they act by different direct modes of action and administered at subthreshold doses, can create unique and enhanced consequences on functional outcomes that are not seen in single exposures. As examples are the effects of this combinatorial exposure to phthalates, where single phthalate exposures show nearly no incidence of hypospadias, but the combinatorial exposure showed nearly a 50% incidence in male rats. And multiple frameworks are being established, advocating and attempting to reduce some of the complexity or organize some of the complexity, advocating that we assess the effects of EDC mixtures on fetal health and development across all of the scales of biology, moving from the chemical structure and properties of these compounds, thinking about molecular initiating events, cellular responses, organ responses, and then eventually being able to predict whether or not we will see adverse outcomes at the level of the organism, whether we will see functional translational endpoints. And there's a lot of good work, growing work, looking in vitro at compounds that share the same molecular mode of action and identifying additive and synergistic effects. But when thinking about neurotoxicity, before investigating the mechanistic origins, we were left wondering if we would see unique and enhanced effects of mixtures of low-dose EDCs on translational behavioral outcomes. And we also began to wonder if, given the compensatory mechanisms available to a developing organism, if essentially death by 1,000 cuts is more insidious. We wondered that if multiple hits, even small ones, along a hormonal cascade could overwhelm the neuroendocrine system's ability to compensate, altering pathways and converging downstream on single disease or dysfunction phenotypes, including the development of behavior. Essentially, we were curious about a very simplistic hypothesis, is that developmental exposure to a mixture of endocrine disrupting chemicals, would a developmental exposure to a mixture of endocrine disrupting chemicals produce unique behavioral dysfunction, even if the single EDCs did not alter behavior? So if we exposed developmentally to an EDC that showed no behavioral deficit, would we see enhanced or unique effects of the mixture? To test this hypothesis, dams were treated from gestational day 7 to weaning with either a single EDC or a mixture. For all of the behavioral assays I'm going to talk about today, we use 1 pup per litter per sex per behavioral endpoint. We've done this twice now, which is good because we replicated some of these findings. But the first time we used regular food and the second time we used soy-free food. And we administered these EDCs orally via treats. This turned out to be quite the challenge. It's amazing sometimes in science how the simplest things can be the hardest. But we really wanted to avoid gavage and avoid sugar-based treats to limit stress during pregnancy and limit the increases in blood glucose daily. And so we decided to take a look at the animal literature, and it turns out that wild mice love insects. And so what we were able to do is we were able to actually expose pregnant dams to EDC-laden frozen mealworm popsicles. They love them. They get very excited for their mealworms, and it made dosing pretty easy. What was not easy, however, was actually figuring out where to start. Where do you begin when you're thinking about developmental exposures to single EDCs and to their mixture? Well, we wanted it to be a complex mixture from broad chemical classes. We needed them to be well studied and well characterized, to basically stand on the shoulders of the giants before us that had done dose response curves or had looked at the dose response range for single compounds so that we could pick a dose that would be close to what we had hoped would be a no adverse effect level for behavior. We wanted broad human exposure, and we did want to sort of test this heterogeneous mode of action to see if it can converge downstream on a endocrine axis, but also to see if there would be developmental influences on the adult central nervous system. And so we picked 4 very well categorized and established endocrine disrupting chemicals: atrazine, PFOA, bisphenol A, and TCDD. And we used the rodent literature to identify a dose that would be considered a low dose that had no effect on behavioral endpoints. Unfortunately, not all of our behavioral endpoints were represented in the literature. We really try and use translational behavioral endpoints, as you're going to see in the slides that are coming, that have analogs in both rodents and in human models. For chemicals like bisphenol A, it was increasingly difficult to identify an OL. There is research now showing effects at like 2.5 micrograms per kilogram, depending on the endpoint, depending on the study. So we used human reference dose of 50 micrograms per kilogram per day. The mixture got all 4. And so we're going to start with our assessment of behavioral function. I'm going to tell you about 3 behavioral assays today that we looked at, and each of them has an analog between the rodent and the human. The first is novel object recognition. And essentially this behavioral assay takes advantage of an animal's natural propensity for novelty. We're interested in things that are new. To make something new, there has to be a training and habituation phase. So the first phase is the training habituation phase, where you put a rodent in a chamber with 2 objects, and those objects are identical. After that habituation phase, those objects then become familiar. And then there's a probe phase, either an hour or 24 hours later, where you return the animal to the chamber. One of the objects stays the same, and the object is changed to be the novel object. Essentially, then you can calculate a recognition index to see how much time they spent investigating the novel over the familiar object. This is the same thing we do with the Fagan test of infant intelligence, where we look to see if an infant spends more time gazing at a novel versus a familiar stimulus. And this also allows for the assessment of short-term memory, essentially. Can they remember which one is the familiar over the novel object? So there's this balance between memory and familiarity. But from the very beginning and the habituation phase, not even the recognition phase of this behavioral assay, we saw that our single EDCs were not completely innocuous. All of the males and all of the females in the habituation phase to the novel objects showed a really unique exploratory pattern where there were significant decreases in the time per approach. And don't worry, I'm about to show you some videos, because I always tell my students that the best way to understand behavior is to watch your animals. But there's a reduction in time per approach that for the females, there was actually a compensation where they increased the total number of times they approached the object, such that there was actually no deficit in exploration time for the single EDCs or the mixture for the females. However, this wasn't true for the males. All of the single EDCs and the mixture shows a reduction in the amount of time they actually explored objects at all. And so here's my video. So pay attention to this animal on the left here. I think this gives the best explanation of a long investigation bout. Remember, this is mouse's attention span. 1, 2, 3, 4. 1, 2, 3, 4. And now I'm going to show you a short investigation bout. In this one, watch this side. Both do it, but you can you'll see two different explorations on this side. 1, 1. And so what you saw is like a reduction in the amount of time per investigation that they spent investigating the objects, suggesting some either reduced attention to objects, perhaps fear of the objects, but given that we did look at some other fear-mediated assays like elevated plasmase and we didn't see any differences, we tend to think that these are actually attentional differences. But interestingly, when we looked at the object recognition phase, only the mixed males showed an inability, showed a reduction in the recognition index significantly different from their vehicle male controls, where they were essentially 50-50. There was no recognition, no increased preference for novelty of the novel object in the mixed males. And there was no effect in the females when it came to the recognition index. The second behavior that we wanted to look at that has nice translational validity to humans is actually using an operant conditioning box to train rodents to press levers for reward. You can do the same thing with kids. And the nice thing about operant schedules is that they shape behavior in very characteristic, very predictable, very reproducible ways across species. And so what a fixed interval schedule does, it actually essentially shapes a scallop shape in behavior, they say. So think about studying for exam that's on a Friday. You don't study Monday, you don't study Tuesday, maybe on Wednesday you open the book and then you cram on Thursday. That's essentially what happens with a fixed interval schedule. As the animals learn it, there begins to be this sort of post-reinforcement pause where there's no responding, then there's a slight increase in responding, and then you see a real increase in response rates right before the end or the termination of the schedule. We put these animals on a fixed interval 60-second schedule, and we look to see how their rates of responding differed by treatment group, particularly because in children, we have seen, there's been research that showed that increased response rates on an FI schedule associates with increased impulsive choice in children. And so again, we have a video. We have a female mouse here who's very good at learning her FI schedule. I'm going to start about 20 seconds in for you here. And again, this is a 60-second interval. So you immediately know which lever is the active lever. There are two other levers that are inactive here, and she knows too. She waits more than half the interval to begin pressing. And then as it gets closer to the end of the interval, she really increases her response rates. And then 60 seconds passed, she heard the tone and she got her reward. And so what we found when we put all of these single EDC and the mixed exposed males on a fixed interval schedule for more than 25 sessions is we actually replicated some previous work that showed that the mix in males increased response rates on the FI schedule. The same thing actually happens in females, but only for TCDD exposed females. Interestingly, as we extended this interval to more sessions, BPA alone does catch up at the very end to the mix, showing an increased response rates at the end of the schedule. Additionally, some new analyses by Sam Weisenthal, who's a graduate student in biostatistics, is looking at this increase in TCDD in the females and actually identifying some significant decreases in atrazine as well that may be responsible for some of the antagonistic effects that we see in the mixture on the FI response rates in the females. Finally, and the last behavioral assay I'm going to tell you about today is social conditioned place preference. And this assay is essentially, does exactly what it says. It conditions a preference for place based on social stimuli. So essentially, this is a conditioned place preference castle for children. Imagine I gave a child an opportunity to play with their best friend in the green room, and then in the peach room, they were forced to sit in isolation in a corner. And we did this over and over and over again. And then afterwards, we brought the child in the room and we just watched them to see which room that they would go into. They would run to the room, the green room, where they had the opportunity to play with their best friend, and probably not go to the peach room where they were forced to sit in isolation in the corner. We can do the same thing with mice. We can take them and place them with their cage mate in one chamber. And in the other chamber, we basically put them in social isolation. We do this over and over again. And then we basically put them back in the chamber and we ask them, which room would you like to go back to? But this is a categorical measure. So it's a little bit not very descriptive. So this is an animal that had been conditioned for social preference in the vertical stripes. So you can see that they're in the center and they sort of take a look around. And then they make the choice for the social route. When we look at our data, we see here that for the females, all of the females chose to go back to the social. The majority of females chose to go back to the social room. For the vehicle males, just like the females, 80% of them chose to go back to the social room. But for the mixed exposed males, 80% of them chose to go back to the room where they were in isolation. So we didn't ablate place preference. It seems like they had a preference for the isolated condition. And taken together, what we saw here is that mixed males actually showed reductions in memory that might be associated with some attentional deficits. They show an inability of inhibiting responding on an FI schedule that had been associated with impulsivity in children. And now we see that there are social deficits in these mixed males. And so I just want to also just bring it back to some of the shared physiology. We had this hypothesis that we could sort of target multiple hits along the HPG axis. And given that we saw a lot of male-specific enhanced effects, we wanted to take a look at serum testosterone production from the HPG axis in adulthood. Individually at higher doses, many of these single EDCs have been shown to reduce testosterone in adulthood. And that is what we found. We did see a reduction in adult testosterone in the mixture alone and not in the single EDCs. What's important about this data is that these were in non-behaviorally experienced animals. These were just home cage control animals. This data actually does look different following behavioral experience. What was also interesting is that these males showed increased anogenital distance. Mixed males showed increased anogenital distance compared to vehicle males. Nice internal control. Our males had longer anogenital distances than our females, but the mixed males seemed to have even longer AGDs, which was surprising given the reductions in testosterone in adulthood. And so we took a look, or we started thinking about sort of the development of the HPG axis. And one of the things that's unique to males is this perinatal surge in testosterone that males experience gestationally and postnatally after birth. This occurs in rat, mouse, and human infants. Importantly for rodents, it only lasts a couple of hours after birth. But it has long been known since the 50s to be very critically important in organizing brain development and behavior. And what we see is actually that there was an increase in testosterone at birth of these mixed exposed males. So in conclusion, we exposed pregnant dams to single EDCs or to the mixture, and we tracked the neurobehavioral and neuroendocrine consequences across the lifespan in both males and females. And we saw that there were significant and enhanced effects in the mixed males on behavioral deficits and sort of lifelong dysregulation of the HPG axis. Interestingly, in females, we saw a lot of compensation and some antagonism. And I just wanted to end with some like big picture perspective sort of at the end. And I would sort of be remiss if I didn't make the statement that hormones don't invariably cause behavior. Instead, these relationships are incredibly context dependent. The endocrine system, as I mentioned, is the body's chemical communication system, and it stimulates response to an environmental context. This has been something that's known since the development of the endocrine system, and the word hormone itself means to urge on, to impel, to set in motion. So instead of thinking of it as a causal role, because there's a great deal of variation in tissue competency and variation in the meaning of the signal depending on the environment and the timing. In other words, it's much more important to center the environment and to think about the fact that hormones sort of change the probability that in a particular environment, in response to a particular environmental stimulus, that an animal will behave in a certain way. This inclusion of context, stimulus, and the environment is really important for predicting relationships between hormones and behavior, and remembering that this is not a unidirectional relationship, that the arrow goes both ways. Once you center the environment, you realize that there's an environment across all scales of biology that are really critically important for helping us understand the development of behavior. As a neuroscientist and actually someone raising children, I didn't just take these pictures of children off the internet. These were mine. These are my two youngest. I'm beginning to embrace the comment that it's complicated. To understand how the brain develops requires a nuanced understanding of cumulative risk factors, including genes, the endocrine system, maternal health, and external environmental factors, including environmental toxicants, nutrition, stress, and importantly, behavior, social and cultural influences that guide the current reality that many neurobehavioral disorders are biased in their prevalence rates, differences in the age and onset, and the way that these disorders present. I'll leave you with my favorite quote, and that is, for every scale in biology, it receives its mechanism for the scale below and its significance from the scale above. We have a lot of work to do moving forward to think about the ways that the environment and EDC mixtures can influence organismal behavior, but it's exciting work. And I would like to thank my lab and my technicians and my graduate students. This is hard work to run this many animals through so many different behavioral assays and do developmental neurotox work, and they are superstars. Thanks to my collaborators and to my past and present funding. And thank you all for listening today.
Thank you so much, Marissa. That was great. While we're waiting for our next speaker to pull up her slides, I would like to remind you to start submitting your questions to the Q&A feature available on the menu bar at the top of your window. We'll begin the Q&A session after our next speaker's presentation. And Katie, I will pass it on to you.
Hello, everyone. Thank you so much for the invitation to present today, especially with Marissa. And today I'll be talking about something that we've been working on for a few years now in my new lab, and this is to identify biomarkers of developmental neurotoxicity, specifically reflective of thyroid disruption. And I do work at the US EPA, but myself or any of the authors, we have no conflicts of interest to declare. So it has been known for hundreds of years that thyroid hormones are required for normal brain development in mammals, including in humans. And two of the main thyroid hormones I'll be talking about today are tetraiodothyronine or thyroxine T4 and triiodothyronine or T3. And these hormones are created in the thyroid gland, which is found in your neck, released into the bloodstream, and then actively transported into target tissues where they control cell signaling. So what happens during pregnancy under cases of extreme thyroid deprivation, like severe case of iodine deficiency or severe maternal hypothyroidism, is it can result in what's called cretinism, which is shown here on the bottom left. And that's associated with low birth weight, altered brain structure, and severe intellectual disability in children born to these mothers. However, we now know that thyroid hormone insufficiency, which may or may not be corrected during pregnancy, has now recently been associated with lower IQ and structural defects of the brain, which you can see by MRI, etc. So this can be caused by a variety of factors, but we're really interested in the role of environmental EDCs and how that might affect the thyroid system. So currently, much like Marissa already discussed, we know that there are many chemicals that do have the potential to interrupt thyroid signaling in vivo. These are chemicals like perchlorate, which is combustion used in fireworks and rocket fuels, poly and perfluoroalkyl substances or PFAS, which Marissa also talked about. They're used in stain repellents and also in the process of creating nonstick cookware. They're surfactants. And then pesticides that are sprayed on various crops that we then eat or get into the waterways. And it's really difficult when we're trying to understand developmental consequences of EDC exposure because we have a lot at play when we're trying to consider how it may affect childhood development later on in life. And this is because we're, of course, exposed to a mixture of chemicals daily. We have to consider metabolism and whether or not a metabolite might be more potent than the parent compound. And we also have to think about if that chemical and metabolites can cross into the placenta or cross the blood-brain barrier of the fetus. And then when we layer on endocrinology on top of that, we also have to take into consideration how maternal hormones may be transported across the fetus and how an endocrine disrupting chemical exposure may affect that as well. So for thyroid specifically, even though the fetal thyroid gland does develop, of course, pretty early on in development, like many organs, at term, a significant portion of fetal thyroid hormones do originate from mom still. So there is a point where the fetus does create its own thyroid hormones and is still reliant on mom. So there's just a lot of things to consider, which make trying to understand the consequences of a chemical exposure really difficult. So this project was really inspired by whether or not we can find biomarkers of developmental neurotoxicity that could be predictive. And this was really kind of inspired to me from blood spot screening, which is one of the most successful public health initiatives to date, where every infant born in the United States gets a heel prick, and then they are screened for genetic disorders. And really the purpose of this screening is early intervention, because just like many diseases, including neurodevelopmental disorders, early intervention is really key to getting a better, more healthy outcome in the end. So for genetic disorders, blood spot screening, this includes things like cystic fibrosis, and then there's also opportunities to screen for metabolic health of the newborn. So we really asked the question and thought, wouldn't it be amazing if we could find a predictive biomarker of thyroid-dependent neurotoxicity that we could screen in something like blood? And if we could do that, could we have an early intervention to maybe eliminate an adverse exposure to a chemical, or do some kind of other correction to help that infant have a better outcome in the end? So this is really what this particular talk is going to be focused on. So this idea of a non-invasive biomarker is kind of a high risk, high reward idea. We had no idea if it was going to work or if it was feasible in the least. So our first step was to really use our animal model. And we have a rat of a complex neurodevelopmental disorders that does model a lot of human characteristics quite well. And the one that I'm really going to be focused on today is because we've done a lot of basic science work and a lot of mechanistic work trying to understand this particular phenotype. And this really kind of powered this project. And this phenotype is called the paraventricular heterotopia, which is shown here. And these are adult coronal sections, rat brain, postnatal day 90. And what you see here on your right is that an animal that was born to a mother that experienced thyroid deprivation during the perinatal period, what happens is that clusters of neurons just kind of aggregate in certain areas of the brain. And this is what the heterotopia is called. And this does occur in humans as well, and it is morphological, but is also associated with functional deficits, including seizures. So this rat model does have an increased propensity for seizures, much like humans that have heterotopia. And we discovered that the heterotopia forms because of abnormal cell migration, because again, a heterotopia is just abnormal clumps of neurons. We affectionately call it the blob. So you can just think of it as blobs of neurons. Our animal model of developmental thyroid disruption also has other phenotypes that are functional, but I will not be going through those today. So when we were trying to understand heterotopia formation, again, our question really went back to why are thyroid hormones controlling, what processes are they controlling, and when they go wrong, why are they going wrong? And this was really based on trying to fulfill some agency needs to develop adverse outcome pathways. But again, we really tried to get as mechanistic as possible to understand the biology underlying the observations. So here, again, we really were focused on the perinatal period because much like some of Marissa's work, that was our critical window. So in this image is a postnatal day 8 rat brain coronal section again, and it's just stained with Nissl to show you all of the cells. The red box is going to be where I'm going to be zooming in with this cartoon. So what we found is that the heterotopia was an outcome, but it really was problems in a stem cell niche in the brain. And this stem cell niche is called the ventricular epithelium. In addition to being amazing because it is a stem cell niche, it also happens to be one of the brain barriers, specifically the cerebral spinal fluid brain barrier. And the brain barriers, I'll also be talking about the blood-brain barrier later. Really, they were evolved to keep things out from the brain. So anytime that something was trying to touch a brain cell, so for example, the cerebral spinal fluid, which is an intermediate biofluid, blood, etc., it evolved to keep things from just passively diffusing. So it's highly selective and it turns on very early in development. So as soon as the brain starts forming vasculature, the blood-brain barrier and the cerebral spinal fluid barriers are active. So what we found is that under normal circumstances, the CSF brain barrier, is populated with multiple types of progenitor cells, including what's called radioglial progenitors. And those are shown here in lilac. And those cells normally, in addition to being a progenitor themselves, they help mediate cell migration of other types of neuroblasts. So what we found that under circumstances of thyroid deprivation, radioglial cells lose their normal polarity, which causes cells to not migrate properly and then forms the heterotopia because cells just basically get stuck where they are. We also attributed that the reason that radioglial cells do this is because they lack cell adhesion of the brain barrier. We then showed that in addition to being the CSF brain barrier, this region also contains a really highly enriched blood-brain barrier. So this is just a really big vascular network, which makes sense because these cells are very metabolically hungry because they're stem cells. And so the blood vessels in this region were very highly populated. And given that cell adhesion also controls the blood-brain barrier or how the vasculature forms, we hypothesized that because we're seeing all of these abnormalities in cell junctions, is it possible that these brain barriers were significantly affected to the point where small molecules may leak from the brain out into general circulation? And if this was true, could we detect these small molecules in the blood? Because again, we have this abnormality in how the brain is exchanging things back and forth between the brain tissue and biofluids. And so for this project, we first focused on microRNAs, which are small non-coding RNAs of approximately 18 to 24 nucleotides. So what are microRNAs? And they're interesting for a variety of ways, but very quickly, again, they're small non-coding RNAs, and when they are co-transcribed in the same cell as a messenger RNA that they can target, what happens is a mature microRNA can bind to this messenger RNA, recruit certain cofactors, and more or less degrade that transcript. So they're negative transcriptional regulators. And besides their very interesting and important role in gene regulation, it was found a few decades ago by happenstance actually, is that people were discovering microRNAs in biofluids. And that was very weird because when you're finding these microRNAs in biofluids, they were found to be acellular, which means that they did not originate in the fluid, they're just found in the fluid. And this includes things like tears, saliva, blood, urine, etc. And so they became very quickly, people became very interested in microRNAs because of this characteristic. Because in addition to being found in the biofluids, they were found to be extremely stable and resistant to degradation. So all of us that have worked with RNA in the lab know that this is not a characteristic of RNAs usually, and they're very hard to work with and can be finicky. But the microRNAs in biofluids are not like that. And they think it's because this figure on the left can show you is that they're packaged in certain ways, which not only helps them diffuse from their home cell into the biofluids, but it also helps them being protected from things like exo and endonuclease activity, which basically keeps them intact. So currently in the human literature, microRNAs and biofluids are being investigated as biomarkers of complex diseases, given all of these really interesting characteristics, which could make them good biomarkers, including cancers and traumatic brain injury. So for this project, again, we're really interested in finding a non-invasive biomarker that we could maybe detect in blood that could be a biomarker of DNT or developmental neurotoxicity. So they seem like a really good candidate molecule for us to investigate first. So for our experiment, we took pregnant rats and we dosed them with a drug called propylthiouracil or PTU. And again, we chose PTU for this first round experimentation because we've done a lot of basic science work with it and we knew a lot about what dosing regimen to give and during what periods to give in order to see our phenotypes. So it was a really important first step for us to really understand how is this even a feasible idea. PTU was delivered through the drinking water, dose of 3 parts per million, which is quite low, especially compared to other studies in the literature, because again, we're eventually trying to model other endocrine disrupting chemicals, specifically environmental EDCs. So on postnatal day 8, we actually performed small RNA sequencing of the blood of both exposed and control pups. And PN8 was chosen because that was the window where we observed a lot of cellular abnormalities in our basic science work. And those abnormalities preceded our terminal phenotypes, and terminal phenotypes being permanent ones. So that includes the heterotopia, which we can first discern at approximately postnatal day 14. And we can do a lot of behavioral and other functional work in adulthood, so about PN 55. So again, PN 8 precedes our phenotypes, and that was very important given how we wanted a predictive biomarker. So getting right into the data, this is serum thyroid hormone data. So thyroid hormones in the blood of dams on lactational day 14. So it's the conclusion of the study. And what you can see is that T4 thyroxine was reduced by approximately 50% in the dams. However, T3 was not significantly reduced, and actually animals exposed or treated to PTU had a slight increase, but it was not statistically significant. So this is just to demonstrate that the maternal exposure was not, we did not slam them or anything. This was definitely a more moderate or mild exposure. So what do the pups look like? So we analyzed thyroid hormones in their blood on postnatal days 2, 8, and 14. And similar to the dams, T4 was significantly reduced on all stages tested. However, T3 was also reduced in the pups where it was not in the dams. So it appears that they have thyroid hormone deficits that may be a little more severe. We can also measure thyroid hormones in the brain tissue itself. And thyroid hormones do have to be actively transported into the brain tissue, again, the blood-brain barrier, across the blood-brain barrier, so they do not diffuse. And what we saw is that on postnatal days 2 and 8, both T4 and T3 were significantly reduced in the tissue as well. So this just shows that the pups indeed had thyroid deprivation around the postnatal period, specifically the perinatal period we were interested in. So now we'll be discussing some cell junction data. So all cell junctions, I did talk about them previously, but you can think of them as mortar between bricks. And when you're thinking about the brain barriers, their whole entire function is really dependent on that mortar being very tight and very secure. And so if it does tend to crumble, then things could leak in and out. And that is a marker of blood-brain barrier disruption or brain barrier disruption, depending on which barrier you're investigating. So in this image, I'm going to be showing you some data where we were looking at PCAM1 in red, which will mark blood vessels, and Claudin-5, which will mark tight junctions. Tight junctions being a cell junction that is critical to blood-brain barrier formation and function. And this is on postnatal day 8. So again, the same stage that we will sequence blood from. So in this brain region, it will contain the choroid plexus, which is another brain barrier, the ventricular epithelium, which is that stem cell niche I discussed earlier. And then everything here in black that you see is actually where the cerebrospinal fluid would be located. Anytime that you see red overlapping with green as colocalization, either orange or yellow, that will mark the blood-brain barrier itself. And I'll be showing you images in this bounding box. So ventricular epithelium, because again, that's our stem cell niche that we were very interested in and we have studied before. So what we have shown on PN8 is that the animals that were exposed to PTU as compared to control. So normally in the control animals, you see a really beautiful apical localization of Claudin-5 along that surface of the ventricular epithelium. And that is, again, that's a brain barrier. So the cells have to use tight junctions to stay really tight to, again, keep things from the CSF from getting diffusing across into those cells. You can also see evidence of blood-brain barrier, which is, of course, colocalization of the green and red to make yellow and orange, which is expected in a normal, healthy animal. However, on the right, in animals treated with PTU, you can see a loss of that apical localization of Claudin-5 along the ventricular epithelium, as well as it appears some abnormalities in both PCAM1 expression itself and colocalization of the two markers, suggesting that the blood-brain barrier may be abnormal. I'd like to emphasize that it appears that abnormalities in cell junction are redundant cell mechanism that we are finding. And in our basic science study published in 2019, a different cell junction marker, specifically NCadherin, which helps forms adherens junctions, was also abnormal in this exact same area of the ventricular epithelium along that apical surface. So together, this suggests that both tight junctions and adherens junctions, both of which are critical to the brain barriers, may be abnormal given thyroid deprivation. So now what does our potential biomarker data say? So this is our microRNA sequencing from the blood or sera of pups. and this is a volcano plot. So on the x-axis is log2 fold change, and then your y-axis is negative log 10 p-value. So when you're looking at this chart, all it's going to show you is if exposed animals are different from controls significantly, you'll see markers in the top upper hand parts of the graph here. So what we found very clearly is that in animals exposed to PTU versus control is that there was a suite of small RNAs, most of which were microRNAs that were differentially expressed in the blood of animals exposed to PTU. So this is consistent with our hypothesis that it might be possible for things to leak out from the brain tissue back into the blood, because if that was occurring, you would expect an upregulation, and that is what we're seeing. And we also have both sequencing and PCR data that also show that all of these microRNAs are expressed in the brain tissue of these same exact animals. So we do have evidence that, again, at the same time, same animal, they're also in the brain as well in the blood. And just for biomarker preparation down the road, all of these microRNAs that we discovered are perfectly conserved in rat, mouse, and human, so they have the exact same nucleotide sequence. And very quickly, sequencing is amazing because you get so much data and you can really get a great idea of what's going on in vivo, but it's very expensive, both cost-wise and computationally. So if we wanted a biomarker, we would ideally want to screen for it by something like PCR, which is fast, efficient, and pretty cost-effective. COVID testing, as we all know, a lot of it is done by PCR. So we took our top candidate microRNAs from the sequencing data and did a lot of validation, and we did show that most of them did validate by PCR in the exact same sample. We, of course, also wanted to try and validate further and see if some of these microRNAs could be recapitulated in sexes, because currently we have no evidence that the heterotopia or our cellular abnormalities that we're finding were sex biased in any way. Therefore, we would expect males and females to show similar signatures of the serum microRNAs. And two of them recapitulated very well through all of the testing battery, which included MIR 543 3P and MIR 495, which we shown here. So taking our data like one little step further, of course, we would want to know, so microRNAs regulate genes, right? So we want to know, what are these microRNAs doing? We find them in the blood. They don't come from the blood originally, they're acellular. So what could they maybe do? And we put this into ingenuity pathway analysis, which is a way to take sequencing data or other types of data and kind of see what it does in the context of the literature. We found some really interesting signatures. And when we see what kind of genes these regulate, both of these microRNAs similarly regulate pathways that we were finding in the brain to be disturbed. So this includes endothelial cell function, because again, we are interested in the blood-brain barrier, cell migration. We are interested, and we know the mechanism of the heterotopia is occurring at this time, brain lesions, organization of cytoskeleton, etc. So it's just very interesting that we're having a signature in the blood that is mimicking exactly what we see in the tissue, which gives us a little bit of confidence. So in conclusion, I just have a few more slides. We originally hypothesized that due to blood-brain barrier and cerebrospinal fluid barrier disruption, perhaps small molecules could leak out. And our working model is that perhaps, yes, that is the case. And we have two potential biomarkers that we're very interested in and are working with right now as well. And this is, again, MIR 495 and 543 3P. And of course, eventually, we would like to really put this in the context of environmental EDCs, because we were trying to make a biomarker, discover a biomarker for a purpose, a bigger purpose. PTU is an excellent model because it gives us data to work with, and we can do a lot of mechanism because it's a very clean, elegant way to do things. But currently in my new lab, we've been working a lot with a PFAS called perfluorohexane sulfonic acid, or PFHXS. And this is just to show you that it's a pretty potent thyroid disruptor in vivo. So we dose dams with either 17 or 50 mg per kg. And this is just really quick data in pups to show you that it can reduce both serum T4 and T3 in these animals. So the question is, could these biomarkers be used in the context of something like a PFAS? And we are working on that right now. And we do have some ways to go, but I hope that in the future, originally we were inspired to find a biomarker, and that was really to enhance chemical testing. So this is both in vivo, of course, like in rats. So a lot of chemicals, if they are tested through OECD testing strategies like 421 or 426 for all of the regulators out there, it would be really amazing if we could evaluate brain status from a small volume blood collection, just like we're proposing. Because if you could do that, it's very quick and very easy to screen by PCR, and it's economical. And it would be just a great way to kind of reduce the number of animals that are used in the testing and to get some data very quickly. And of course, and no illusions here, we know that this is very far down the road, but it would be amazing if we could screen people that are living in polluted areas, specifically through a newborn screening method, much like that is already in progress today. Because if you could do that, it would be potentially possible to, again, remove an exposure or do some type of intervention early on in blood-brain barrier disruption before something could happen to the child that may be more permanent. So early intervention is really key. And I'd like to thank everybody for listening, and of course my co-authors. And just to mention that this work was done by 3 postbacks and myself for the most part, so I have to give it up for them. 2 of them are in grad school and 1 just started. And we hope to publish this work soon. We're actually doing some wacky blood-brain barrier experiments as we speak, so hopefully it will be published shortly. And I'm happy to take any questions. Thank you.
All right, thank you, Katie and Marissa. Those were great. So if people have questions, you could put them into the Q&A box down here. We have a few minutes, a little less than 9 minutes. I don't see any questions yet, so I'm going to ask if you guys have any questions for each other, first of all. Put you on the spot.
I have a question for Marissa.
Yeah. Go ahead, and I'll ask you one too.
Okay, are you going to be doing more mixture studies with PFAS?
Yeah, I think I think that there's sort of 2 aspects to this work. One is, I think we've really sort of understudied this early perinatal surge in testosterone, whether it's this mixture or other mixtures. So one of the things that we began looking for is, did anyone see this sort of signature of reduced testosterone but increased anogenital distance? And there's some really interesting stuff by Andrea Gore and others that did some PCB mixture work. And so we're sort of taking a look at that and, preview to preview, seeing some elevations in testosterone early. And so we want to sort of get an idea of how rare is this? How common is it? And then definitely, there are so many new perfluorinated compounds out there. And it's clear that there are mixtures of perfluorinated compounds that humans are exposed to. And so I definitely think looking at combinations of perfluorinated compounds, even by themselves, could be very interesting.
Yeah, definitely. I agree. And yeah, that was, I think, one of the most interesting parts of your data is that disconnect between the anogenital distance, like what you'd expect potentially versus what you saw. So it
turns out it's way more complicated. I'm not a reproductive toxicologist, but there's a lot of, DHT plays a really big role in other compounds. But yeah, I think it makes a complication in thinking about something or thinking about these compounds in vivo. It's a lot harder to call them sort of androgenic or anti-androgenic.
Yeah,
because the systems are more complicated than, for example, just an androgen receptor binding assay.
Yeah.
Has anyone looked at your microRNAs, Katie, to see if they are like associated with other disorders? I mean, the goal, it's amazing actually that they're stable and that you're able to measure them. And the response was so robust with the thyroid disruption.
There have been a few cancer and TBI work, and a lot of the cancer work, cancer is also very interesting because it's a lot of times abnormalities in cell adhesion and the ways cells are communicating, which can then lead to metastasis and whatnot. So it is very odd, but other people that looked at the microRNAs in tissues, they do see just highly similar pathways, even if it's in a different tissue, if that makes sense. Yeah. But yeah, unfortunately, not a lot of people study the brain, except traumatic brain injury is the main one. So.
All right, let's move on. We've got a bunch of questions now. Katie, did you measure TSH changes and what did they look like?
Oh, Ms. Marika, hello. Yes, we did. We recently did actually about a month ago, and the dams have a slight increase in TSH, which was statistically significant. But But I will say that the same exact exposure paradigm, we've done it several other times, and sometimes TSH is up a little bit, sometimes it's not. So that wasn't too surprising, but I would personally still put it near the hypothyroxanemic range, so pretty mild. And in the pups, they did have elevated TSH at postnatal day 8. And that wasn't too surprising given that they seemed definitely more affected than the mom.
And also, do you think there may be other pathways that could be affected by the hypothyroid status of the moms, aside from the CSF barrier being targeted?
Yes. One of the ones that's pretty interesting is there definitely seems during hypothyroidism, there's a change in the shift of excitatory and inhibitory neurons in the brain, so that like differentiation pathway is abnormal. And that we do think, my postdoc mentor, Mary Gilbert, especially thinks that that can cause abnormalities in things like synaptic transmission and some of the behavioral deficits. That is a little more difficult. We don't have quite as much mechanistic work done on that. So that wasn't the focus of this particular project. And that actually happens later in development too, compared to what we studied here. So, but it is very interesting. And it does appear to be very robust and reproducible across laboratories.
All right. Marissa, why did you choose the BPA dose that you did and not lower? I think you mentioned that a little bit, but it looks as if testosterone levels were higher in BPA treated males, although maybe not significant. And Ted says he might have misread the slides.
Yeah, so it wasn't significant. And yeah, BPA is a hard one. So there is a lot of discussion about low dose BPA effects and the non-monotonicity of those effects. And so really when we came down to it, we decided the question is sort of as, are these low doses safe? And is the mixture cause an enhanced effect? And so we decided to go with human reference dose, the dose that is currently considered safe. So that is why we chose 50 micrograms per kilogram and not lower. That did have effects, right? That did have a consequence for us because although testosterone was not significant, when we let the FI go out longer, we did see single effects of BPA where it eventually caught up to the mixture. So the mix showed deficits in FI response rates, elevations in FI response rates right from the beginning, straight out the gate, but eventually BPA did catch up. And so, so I do think there were consequences to choosing 50 micrograms per kilogram, but there's a lot of discussion in the low dose BPA range about, you know, one of the hardest things for mixture studies is how do you put them together and what doses do you choose? Ideally, we would have a dose response curve for every single one of these compounds. But again, the FI behavioral assay that you saw, that required 120 animals to run that behavioral assay. There's 6 treatment groups. We have at least an N of 10 and we have 2 sexes. So it actually was even more. Luckily, we have a really amazing behavioral core here. So we had 30 operant chambers, but it's really quite a bit of work. And so, you know, doing the dose response is hard, but ideally, yeah, I'd love to know what happens at lower doses.
Yeah. Someone else asked, have you considered looking at mixtures in a class like phthalate mixtures, PFAS mixtures? Or, I mean, you know.
Yeah, so it seems like the Aerochlor 1221, which is a PCB mixture, does influence testosterone early. And we would love to do the same class of, like a class of perfluorinated compounds and take a look. I will say this is traditionally how a lot of mixtures work is done. You know, thinking about dioxin equivalency factors and thinking about PCBs is thinking within the same chemical class. And although I think that work is super important, it is also true that we are exposed to compounds from really diverse chemical classes. And I am increasingly sort of wondering if and when really you might see sort of downstream convergence if we pay attention to the physiology of the organism and the function of the organism, that it would be beyond just the same chemical class, that we would see some of these mixtures effects with broad chemical classes as well. So I think both of the work, you know, all of that work needs to be done. More work overall needs to be done on mixtures, I think so.
Katie, seems like the blood-brain barrier disruption may set the stage for enhanced susceptibility to many exogenous neurotoxicants. Could or should we make efforts to stratify neurodevelopmental epidemiology studies by microRNA evidence for BBB disruption, if that makes
Yeah,
sense.
I think eventually for sure that would be amazing. Like I said, there's not a ton of human data for blood-brain barrier disruption microRNAs that I know of, except for traumatic brain injury, which does create blood-brain barrier disruption, and maybe a few epilepsy studies. But again, those are very obviously focused on certain traumas and not necessarily about a chemical exposure. But yeah, I think that would be amazing because, as you mentioned, soon as the blood-brain barriers opened up, potentially other things could go wrong. And I also even just think about how, for example, stem cell niches, how are they functioning any differently? Because again, glucose is not diffuse, something as simple as that. So like getting things in and out are extremely important and highly regulated. So anytime that is not normal, I think the effects of that are unclear, but potentially very scary or could have a lot of consequences. So I do hope, especially in toxicology, environmental toxicology, we can have a little more love for the blood-brain barrier because it is so important to just normal function and development. And, you know, it is soon as vasculature is present in the brain during fetal development, so extremely early, it is on. So that, I think, shows us evolutionarily how important it is.
And then someone real quick wonders if you plan to test the biomarkers with other chemicals, especially those that can cause high thyroid levels.
So for hyperthyroidism, I personally haven't worked much with hyperthyroid type chemicals in vivo. All of the ones that we've worked on and I've been working on now always lower, but for sure higher. To be honest, I'm not really sure what would happen to the blood-brain barrier because I've never tried, but that would be very interesting because we do know that hyperthyroidism during pregnancy is also leads to neurodevelopmental abnormalities. So that would be a very good future study for sure.
Okay, and then there's only one more question, and basically it says that females are, testing higher in intelligence tests and that sort of thing, and they're wondering if the difference between males and females could be a response to ubiquitous environmental chemicals. Maybe some of the difference, that's sort of a hard question to answer.
That's okay. I think I'll take a shot. I think that's one of the reasons why it's really so important with things that are not translation, that are harder to translate behaviors, like things like maternal behavior and intelligence and aggression and empathy. These are incredibly culturally determined behaviors. This isn't like we're talking about motivation and impulsivity and attention. And so one of the things that you have to remember is that really for a long time, sexism has kept women from showing up and from, really being able to engage in a lot of these things. And I think that there's never going to be a circumstance where one sex is smarter than the other. Overall, I think you're really just seeing shifts in cultural patterns across time. And I think that that's really a question for culture and society.
All right. Well, I think we've got it all. We're at the end of the hour. I'd like to thank you both for speaking. It was really interesting. And Hannah, I'm going to turn it over to you.
Thank you so much, Katie and Marissa, for this really interesting talk. We're approaching the end of today's webinar. A video recording will be available on CHE's website soon, and tomorrow you'll receive an email containing a link to the video. The next CHE Alaska Partnership webinar, Protecting Water Quality, Health, and the Environment, Urgent Questions Pertaining to PFAS, will take place November 11th. Then on November 17th, we will hold our next CHE EDC Strategies Partnership webinar titled Mechanistic Mediation of Flame Retardants in Preterm Birth. You can find details on our website at healthandenvironment.org. If you are new to CHE and would like to stay updated about upcoming events or more, please sign up to receive our newsletter by selecting the Join Us tab at the top of any page on our website at healthandenvironment.org. Additionally, if you appreciate these CHE partnership webinars bringing you the latest environmental health research for free, we encourage you to support CHE's ongoing work by making a tax-deductible donation via our secure website. Again, our website is healthandenvironment.org. With that, I would like to thank our speakers, Marissa and Katie, for your time today. Thank you so much for presenting. And to you, Sarah, for your excellent moderation. Wishing everyone much health and wellness. Take care, and we'll see you soon.